All right, so once you get signed in, guys, go to Google Classroom, and you will find that in the stream I have placed the microscopy lab assignments, okay, or you can go into the classwork tab and find it there as well. All right, right now what's in there is the PDF, which is just the instructions. That's what I'll be working off of, so you don't have to have that open because it'll be up on the screen, okay? And obviously your Google Doc, which is what you should have open so that you can start doing the pre-lab stuff with me here, okay? And there's a couple of videos that, well, this is the observations one, so you don't need that for today. The cell theory and lab diagrams is what also I'm going to go through today, how to do a proper lab diagram, okay? So you don't need to watch any of those right now. It's mostly for people who are absent, okay, who will need to see that. Okay, so you should have your Google Doc open at this point, and that's where we'll work from. So, purpose of the lab on Monday is twofold. We want to gain experience using the microscope, okay, because a microscope is not a piece of equipment that most people get to use on a regular basis. It's a bit finicky, okay, and so we need to get some practice using it. And secondly, to kind of be able to observe and understand how it works. All right, then that's kind of the lab report part of it, okay, is how does the microscope work? How does it allow us to see tiny specimens, which is what's written here as the problem, which of course needs to be rephrased in your own words in the form of a question. Okay, so generally we are looking to investigate how a microscope works. Now we talked about this on the first day of the unit on Tuesday, okay, when we were talking about Hooke and his first microscope that he built, right? So in general, and this will be helpful when you're doing your hypothesis and your analysis and your conclusion and stuff, okay? In general, a microscope works like so. It's still not working. What is going on here? All right. So generally, a microscope works like this. Whoop, you have a specimen, okay? And let's say it looks like this, right? You have a series of lenses, which I'm just going to simplify to be a single lens, so a curved shape, or a curved piece of glass, right? And then you have, that's an eye. I'm not an artist, clearly, okay? All right, so here's what happens, okay, with a microscope. Light, a light source underneath your specimen, okay, causes light to pass through the specimen and hit the lens. Okay? When it hits the lens, light bends. It's a property we call refraction. Okay? Whenever light encounters a new medium, so in this case it's going from air to glass, okay, its speed changes. And because the piece of glass is curved, its speed and direction change. So glass can bend light. Okay? It's why we use it to make prescription glasses and things like that. We can change the direction light travels so that it turns to a different place. So what happens here is the light that hits the lens head on just goes straight to the eye. Okay? The lens that hit this, or the light that hit this side of the lens is going to be bent and it's going to go this way. Is there something wrong with that one, Brandon?
Yeah, it's not loading.
Can you put it on the top and then I'll have a look at it? Okay, and then the light that hit this side goes over here, right? Now what that leads to is this divergence of the light. So the light that came from this side of the specimen ends up over here, okay? And now, I shouldn't have drawn that eye. Now it's going to be confusing, okay? So now, the light that came from this side of the specimen ends up over here, okay? And I get the specimen being inverted and bigger, okay? So the thing, that little circular thing that was on the left in the specimen is actually going to be on the right when I look at it, okay? Because the lens is going to flip it during the process of magnification. Everybody all right with that? Okay, so that's in general, very basically, how a microscope works. Okay, lenses bend light, and that results in its magnification. Right. You can also have lenses that converge light. Okay, like at this point right here. Okay, if you ever like took a magnifying glass and like caught the sun with it, you can get a spot that's really, really hot where all the rays converge. It's the same idea. That would be this point right here where all the rays are converging, and the light would be very intense there. Of course, you've never burned any ants or anything with the magnifying glass because nobody would be that mean. Okay, so everyone all right with that? How that works? Okay. So for our design here, for our experiment, this is how the experiment is going to work. You are going to be looking at 4 different specimens. Okay? The observations you're going to make about each one of those specimens, okay, is going to be kind of different because obviously each specimen looks different. Okay, so you're going to have a lab diagram of each of the things that we're going to look at through the microscope, and what we're really trying to see here are what are the properties of an image after it goes through the microscope. Okay, so our main observations are what are the properties of an image once it's gone through a microscope. And then, obviously, as we're looking at all the specimens, we're going to try not to change microscopes right in the middle of the experiment, or change our lighting significantly, or basically anything that could affect how images look through the microscope would probably need to be controlled. And I would say that that would include the device we're going to use to take your pictures. Okay. I would use the same device each time. Okay, for your hypothesis, as always, we want that to be an if and then statement. Okay, the if part of your hypothesis should tell me how a microscope works. Luckily for you, some thoughtful person just gave you an explanation of exactly how that happens. Okay, so the if part of your hypothesis should tell me basically how a microscope works. Part, as always, describes the experiment. 4 specimens will be observed through a light microscope. Okay. The then part what are the characteristics of the image that you will see? And so if a microscope works by doing this, and specimens are observed under the microscope, then the images will have the following properties. And you can list them. Okay? I would say it wouldn't be a bad idea to work in a because in there somewhere. And the properties will be like this because. So the procedure is pretty straightforward. There are 4 specimens that are going to be observed. Okay? but we have to do it in the right order. We want to start with something fairly large and fairly easy and fairly familiar, okay? And then we're going to move to a large microscopic organism, okay? A smaller microscopic organism, and then an even smaller microscopic organism, okay? To kind of challenge ourselves with the task of focusing and things like that, okay? So the first specimen that you're going to look at is the letter E. Okay? Now, in order to look at the letter E, you're going to make what's called a wet mount. Right? A wet mount is you're going to make your own slide. Right? So I will have blank slides, which are just a rectangular piece of glass, okay? And they will be at the front of the room. So you'll get this rectangular piece of glass. And what you will do is you will cut out from a newspaper a lowercase letter E from an article, not from a headline. Okay, we want something small, not something this big. Okay, so you're going to have this small piece of paper, okay, and on it will be a small lowercase letter E. Okay. What you will do is you will take that and you'll put it on the slide, like so. Then you'll take an eyedropper, okay, and there'll be a bunch of eyedroppers and a beaker of water on the front table, okay, and you'll put just a couple of drops, 2 at the most, okay, 1 is often good enough, onto the letter E. Now, while you're doing that, my suggestion is that you're holding the letter E down on the slide with a pencil, okay, because if you don't, the polar nature of water will cause it to bounce and sit on the top of the bubble, okay? So we don't want that. We want it to stick there and soak up the water and then stick to the slide, okay? So you'll put a drop of water on there while you're holding it down, and then you will take a cover slip. And a cover slip is just a small, clear, square piece of plastic, okay? And you will take that cover slip and you will set it on the slide and drop it onto the letter E. Now, that looks like this. So here's the slide. So we're looking at it kind of from the side, okay? Your letter E is on here. Your letter E is on here already, okay? You've put your drop of water on there, okay? And now you're going to take your cover slip and you're going to set it on the slide at an angle like this, and then just let it go. And it'll fall down, squish the water to the edges, and it'll stick. Okay? That's the whole reason we use the water, right? If we just set that on there with just the letter E, is there anything that's going to hold it all on? No, and then even like you're breathing while you're looking through the microscope will cause it all to fly off. Okay? So the water is going to be there, okay, in order to make this a true wet mount and keep everything in place. Okay? So that'll just fall down, it'll squish to the sides, you got your wet mount, you go and stick it in the microscope. Okay, now here's the key. When it goes under the microscope, it needs to be oriented like this. So when you put it in, make sure when you're looking at it and you put it in that it's right side up when you look at it. Will it be right side up when you look through the microscope? What happened with this image? Okay, so it's okay, but like it's going to look different, and that's what we want. That's one of the things we're supposed to be observing in the lab is what happens to the image as it's magnified. That's going to be one of the things. And the letter E is about the only thing we look at where you can tell if it's right side up, backwards, mirror image, inverted, whatever. Okay, so that kind of makes sense? That's the whole reason we look at the letter E. I look at an amoeba, I don't know if it's upside down or backwards. Okay, amoebas are blobs, and they look the same no matter what. All right, so that'll be how you make your letter E. Once you're done with the letter E, you take the plastic cover slip and the letter E and you throw them in the garbage and you bring the glass slide after you've washed and dried it back up to the front. Okay, super easy. Then we'll look at an amoeba. Okay, so we'll look at the letter E Then, so this will be number 1, okay? This will be number 2, this will be number 3, and the Euglena will be the 4th thing. You don't have to make wet mounts of the other 3. They are already prepared slides. So you just have to grab one slide and go back to your table and have a look at it under the microscope, okay? So you're going to take pictures of each one of these specimens, okay, with your phone through the microscope. The letter E, you're going to take a picture of it on scanning power. Okay, that's the small red lens. Okay, if you go any bigger than that, it takes up more than the field of view. Okay, you won't be able to fit it all in there. The Amoeba, you're going to take the picture of that on low, because it's the second biggest one. So that's the yellow moderate length lens. The Paramecium and the Euglena, you'll take both the pictures of those on high power. Okay, so as I said, we're starting with something big, and then we're working towards the smaller stuff. Okay, questions on that? All right. So what we need to do with those pictures is we need to make a proper lab drawing or lab diagram, because you guys don't actually have to draw anything, right? A proper lab diagram looks like this. Okay, so your name and the date go in the top right-hand corner. Your picture kind of goes center left, right? And then that leaves room for you to make all of your labels out to one side, okay? Because we don't want our lab diagram to look like the specimen is exploding, okay? Because sometimes people do that. They put the picture in the middle and they got lines coming out of it in all different directions, and it literally looks like the specimen's exploding, okay? Which is not very tidy, right? So we want all of the labels to be off to one side, and we want them all to be straight lines, okay? They should come out and end horizontally. Sometimes you're going to have this situation here, okay, where two structures are too close to each other for you to make horizontal lines for both. So you go up diagonally or down diagonally for one, and then over horizontally. Okay, it's again just neat. That way all the labels end up on the same side. All right, everybody good with that? And then at the bottom, you will have the name. So this is an amoeba. Okay, you'll look at one that's, I mean, they all are kind of blobs. Okay? They're generally like this. The magnification you viewed it at. Okay, this is actually high power. You're going to view it on low power, so it would only be 100x. And the approximate size. Okay, do you remember how we do that? Okay, so if I'm looking at an amoeba on low power, okay, the distance across on low power, we wrote this down the other day, is 1,600 micrometers. So my amoeba might be about that big. Okay, takes up about 1/4, maybe slightly more than 1/4 of the field of view. Okay, 1,600 divided by 4 is 400 micrometers. This is, I wrote 450. Okay, ringing a bell? All right, so that's what your that's proper format for a lab diagram. It has to include all of this. The title's at the bottom. That's counterintuitive. Usually titles go at the top, not on a lab diagram, goes underneath. Magnification and size. Okay, that's what a proper lab diagram should look like. All the labels to one side and your name and the date in the top right corner. Okay. How big should your lab diagram be? It should take up a full page, each diagram. So in your Google Doc, when you do this, each lab diagram should take up a full page in your lab report. Okay? Because what I hate seeing is I'm reading through somebody's lab report, okay, and they do this. Like underneath all of this stuff, they're like, and I'm supposed to know what that is, okay, because it's literally a thumbnail, okay, instead of something I can actually see. Right? So make sure it takes up a whole page, each one. Okay, questions on the format? Okay, here's the way to build it, because I understand that not everybody has, like, you know, image rendering software at home or anything like that. Okay, this can all be done within Google Docs and Google Drawings. Okay, so what I'm going to do is open up the lab report and show you how this works. Okay, so I've got my lab report here, and I want to put in my lab diagrams. So it says here, observation, see lab diagrams on following pages. Okay, so I'm just going to go and put it kind of underneath everything. Okay, and I'll just put in a page break here. Oh, where is it on here? Break, there it is. Page break. All right, so now I've got a fresh page. Now what I want to do, and you guys can follow along here, okay? I'm going to go to Insert, Image, okay? And I'm going to go to, oh, sorry, Drawing, sorry, Insert Drawing, okay? And I'm going to go to New. All right, so I'm going to actually make a Google drawing. So it opens up possibly the world's worst image program. Okay, but it's okay. We're just putting labels and titles on our picture that we took, right? So the next thing I would do is I would insert a picture. Okay, so I hit that little button there, okay, and it gives me these options here from your albums, from your Google Drive. Okay, for me, I'm just going to upload it because I put it on my desktop so I can find it quickly. You will probably have to put it in your Google Drive to access it that way, okay, when you're doing this. So I'm going to choose an image. Okay, and I'm going to go to my desktop, come on, where I put the Amoeba. Okay, so now my Amoeba is in there. This is probably not the best orientation for it, okay, so I'm just going to bring it down a little bit so that I can flip it. So, bring it over so it's more to one side. Okay, now I've got my picture in there. Now I want to insert some text. Okay, so I want to put in like my name and the date. So I go to the text box and I click here and I just click and drag till I get the box, and then I can type in my name and the date. So now I've got that. Okay, and then I would go underneath. I do the same thing to put my title and my magnification and my size. So actually for us it'll be 100x because we're viewing it on low power, and 400 micrometers. You can just use U, okay? Don't go looking for the mu symbol, just use U. Okay, we talked about that the other day as well. All right, and then I'm going to start labeling my structures. All right, so to label my structures, I'm going to use the line tool. Okay, so that's that thing right there. Okay, and I just want regular lines. And I'm going to go OK. And I try my best to make sure that they stay horizontal and that they end at about the same place each time. Okay? And if I need to do that thing that I showed you, okay, I would go up and then over. Okay? Everyone following me there? Okay. And then put a text box on the end of each one to tell me what it is. Right, so that would be a water vacuole. And done. Okay, that's all there is to it. That's all there is to making a lab diagram. Okay, so now that that's done, okay, I click Save and Close, and it'll put it in my lab report. Okay, and it took up pretty much the whole page. Okay, I can always make it bigger if I want to. Okay, but that's the size it spit out. It's kind of formatted to that page already. I can't make it too much bigger, but it's big and easy to see. All right, everybody good with that? Okay, so you don't have to have any other programs. You can do it all within Google Draw, okay, and use the pictures that you take. All right. Now, for this lab, I get that we will not have gone over the parts of the cell yet. Okay, so I don't expect that you would know what all the things you're looking at are. So on this lab, you can just label things whatever you want. Okay, so big brown dot outside of the cell. Okay, like that's fine for this lab. The next lab we do is all about the anatomy of the cell. Then I'll expect the labels to be right. Okay, but obviously I can't expect you to go out and know what all those parts are if I haven't taught you that yet. Okay, so for this lab, you can call it whatever you want. Okay, you can call it the poop bubble. I don't know, whatever you think it is. Okay, that's fine. Okay, all right. Everyone good with how the lab drawing works? So. For your analysis, okay, there's only 3 questions. They're pretty straightforward, okay? Using what you saw under the microscope, in particular, the size, the orientation, and any other general characteristics, explain what you think happened to enlarge the specimens. In other words, how does a microscope work? Okay, that would be question number 1, but with some justification. For number 2, describe and explain the steps you took to find and focus your paramecium, which is the 3rd thing you're going to look at. So that's pretty straightforward. It's like, I got the paramecium slide, and I put it under the microscope, and then I did this, and then I did this, and then I did this. Just walk me through the steps you used to get to the point where you could take your picture. Okay? And then for number 3, okay, we talked about making the wet mount. I talked about, okay, putting the water on there. Okay, I would also say that part of this answer, okay, has to do with something that water has or does or is that other things might not be, you know, like okay, there's a reason that we do that. Right? You might be able to explain using some of the stuff you wrote in that answer on your unit exam, right? Why the cover slip stays where it does and all that kind of stuff. For your conclusion, same as always, copy paste your hypothesis, and then, based on what you saw through the microscope, the size, the orientation, etc., okay, did the microscope do what you said it was going to do? Did that enable you to see the tiny specimens? Okay, can you accept or reject? And then, sources of error, right? Anything that got in your way, okay? Anything that could be improved that wasn't human error, right? I would even say that for this one, and this is going to sound like it's human error, but it's not, that inexperience could be a legitimate source of error for this one. All right, so this slide here, guys, is just to kind of reiterate, how do we estimate the size of an object? Okay, so if I want to estimate the size of this little thing here, okay, this guy here, I'd say that 1, 2, 3, 4, 5, 6 of them fit across. So I'd go, you know, divide the field of view by 6, and that would be the approximate size of that specimen. If your specimen is long and skinny, which dimension should you do the estimate on? The long or the small? The long, always on the longest. So if your specimen is noticeably longer in one dimension than it is in the other, always do your estimation on the longest dimension of it. Okay? For some of your stuff, it's going to be pretty much the same no matter which way, so it doesn't matter on that. But for things like the Paramecium and the Euglena, okay, they do have a longer dimension and a shorter dimension. Always measure the longer. For the letter E, it's practically a circle, so it doesn't really matter which one you pick. Okay. All right. Oh, see, that was a hand-drawn one. That's much worse. Okay, things to remember. Your field of view for your scanning power lens, and I think you guys wrote these down the other day, is 4,000 micrometers. Okay, so that means when you look through the scanning power lens, the distance from one side to the other is 4,000 micrometers. Okay, on the low power lens, the distance across is 1,600 micrometers. Okay, and on the low power, or the high power lens, sorry, it's 400 across. So, okay, when you're estimating the size of your objects, remember that, okay, so you can get a fair estimation. Okay, so what we see as we go up in magnification is that the field of view decreases, but the detail we see increases because we're magnifying it more and more and more. Okay. Everyone all right with that? Yeah? So if I was looking at the letter E, okay, on my microscope, okay, it would be something like this. Okay, on my scanning power lens, I see the whole E. Okay, but when I switch to the low power lens, I'd only see that much of it. Okay, I'd see it in more detail, but I'd only see that much of it. If I moved it up to high power, I'd probably only see that part of it, but I'd see it in a lot of detail. Okay, everyone kind of following that? Oh, there was my wet mount. Okay, so these are the specimens that you're going to look at. I apologize, the resolution didn't upscale on the new board. So your amoeba, depending on what slide you get, we have slides from different manufacturers of the amoeba, and so they're stained differently. Okay? Most of the slides, the amoeba is going to appear a light blue color, okay, with a kind of a pink nucleus and things like that. And there's a few of the slides that will have this stain where it's more of a pinkish purple. Okay? Generally, amoebas are a blob. Okay? Sometimes they look like a starfish, okay? But most of the time they're just kind of a blob, okay? And they can have any shape, right? But you'll know one when you're looking at it. Okay? The third thing, that's the second thing you're looking at. The third thing you're looking at is a paramecium, okay? So this is the paramecium here, okay? And they are kind of hourglass shaped or kind of like that. So they're longer and skinnier. If you look really closely, you can see around the outer edge, all these little things here. They look like hair under the microscope, but they're actually called cilia, and it's what it uses to move. Okay? These specimens are all preserved, so they're dead. Okay? So they don't go anywhere. But a paramecium, if you're trying to look at a paramecium when it's alive, you have to put like glycerin, which has the same consistency as pancake syrup, okay, onto the slide to slow them down. Because otherwise they just go flitting across the field of view because they're really, really quick. Those cilia wave back and forth and they can just motor. So if you put the glycerin in there and then they get all like gummed up and they can't move very well. Okay, they're going to be pretty easy to find. The stains are different. Again, we have slides from two different manufacturers. On one of them, they'll all be the same color and it's kind of the bluish color. And on one of them, you're going to find some that are blue, some that are green, some that are brown, some that are purple. Okay, and that's because paramecium, their color is based on what would be the equivalent of their gender. Okay, but paramecium have more than two genders, so to speak. It's not, gender's kind of an analogy. It's not really a thing, but it's kind of like that. Because they, before they divide, can actually exchange a small little nucleus that has some genetic material in it. Okay, and that can allow them to have genetic diversity instead of making an exact copy of themselves. Okay, they can exchange DNA with another paramecium, and then their offspring would be slightly different. Okay, so they have different colors because of that. And then the last thing you're going to look at is the smallest thing, and it's not going to be green. All these slides are stained the same. It's kind of a bluish, faded blue color, and the Euglena is really small. On your scanning power lens, you'll barely see it. Right? It'll look like a tiny little blue sliver. power, it'll be obviously slightly larger with a little bit more detail, but still kind of a sliver-shaped thing. When you get it on high power, okay, you'll be able to see just the tiniest bit of detail, but they are quite small. All right, so they will be the biggest challenge for you on Monday to actually get focused and see. All right. Now the Euglena is kind of an interesting organism. It's mobile and photosynthetic. So it's a plant that can move. Okay, so it's full of chloroplasts. Okay, it's the organelle that carries out photosynthesis, but it's got a long whip-like tail, okay, that it can just whip back and forth and move pretty quickly. Okay? So it actually, when it moves, it swims kind of like a sperm, if you've ever seen what those look like when they're moving. Okay? They just have a long whip-like tail, okay? And that's how they move. Okay? All right, that is what I've got to tell you about the lab reports. So we've got another, 8 minutes or so here. Oh no, sorry, we got more than that. We got like 12 minutes. So you can work on putting some detail into your variables, into your hypothesis, okay, that kind of stuff here for the remainder. Okay? Any questions? You may. Any other questions?